In this video we're going to have a closer look at indicators. An indicator is a chemical substance that changes colour depending on whether it's in an acidic solution or in an alkaline solution. Now the structures of these indicators can be quite complicated. For example, phenolphthalein looks like this, where it's got 3 aromatic rings, a couple of alcohol groups and what's called a lactone in the centre. What you'll usually find is that indicators are coloured solutions. That means that they absorb light in the visible spectrum of light. The thing about phenolphthalein is that it goes from colourless to very, very light purple colour, depending on whether it's an acidic solution or an alkaline solution. So if it's colourless, that means our eyes can no longer pick it up. That means it no longer absorbs light in the visible spectrum. What you find actually is a lot of compounds that have these aromatic rings connected in a certain way, or even have double bond followed by single bonds, etc. What you find is they absorb light in the visible spectra. Okay, so aromatic rings absorb light in the UV range, and then if we introduce aromatic rings close to each other or double bonds close to each other, what you get is a system which moves into the visible spectrum, so we can see it. So that's different chemistry, but nonetheless interesting to know. So there's phenolphthalein. Other indicators from the lab, we know methyl orange is another structure. Litmus is another indicator. I won't bother drawing them out. You don't need to know them. You know that indicators such as universal indicator, for example, is just a mixture of indicators added together, and we can get indicators which are natural substances. Most are dyes, colour stuff that we see. Like you can get ones from red cabbage and things like that. But the important thing to note is that it changes colour depending on if it's an acid or an alkali. And most of the indicators that we come across, for example, are weak acids. And we know that weak acids dissociate to a certain degree. So I can represent it like in a weak acid. So the acid itself will dissociate to a certain degree into H+ ions and its negative counter ion. So I've just changed the arrows there just to show that it's in equilibrium, okay? Think about an equilibrium, go back to the equilibrium video that I looked at before, and you can see that if I add acid to this solution, what's going to happen? Adding acid to this solution will increase the H+ ion concentration. What will the system do? Well, according to Le Chatelier's principle, the system will look to oppose the change and so increase the concentration of Hn. Now, because all of these indicators that I've mentioned here have different parts to them, they will dissociate to a different extent. So therefore, they will change colour at a different pH because they've got a different acid dissociation constant. So I'm going to introduce a new term here, and it's called the range. Now the range is basically just the pH range, or the pH interval, over which that indicator changes colour. So what we know is that they'll each change colour over a different range. Phenolphthalein changes colour in the alkaline range, about 8 to 10. Methyl orange changes colour between around 3 and 5, and litmus changes colour between around 5 and 8. So briefly, just to recap the colours, phenolphthalein is colourless in acid and pink in alkali. Methyl orange is red in acid and yellow in alkali. So you may notice that the methyl orange is actually a difficult colour change to see when you do it. Originally it looks a bit like an orange colour. The litmus, red in acid and blue in alkali. Okay, you may wonder why the range is important, because surely you can use an indicator for any reaction, but you can't. There's 2 things to note here. One is the end point of the chemical reaction. The end point is essentially when the indicator has changed colour, and the equivalence point is the point where the volume of one solution has reacted exactly with the volume of the second solution, taking into account the stoichiometry of the reaction, i.e. neutralization. So we need the end point to be as close as possible to the equivalence point for us to be able to see the colour change. Now, to find an equivalence point for a reaction, essentially what we need to do is we need to carry out a reaction, use a data logger to monitor how the pH changes as a base is reacting with an acid. So we're going to get different titration curves for different types of reaction depending on whether we've got strong acid and strong alkali, strong acid, weak acid, strong alkali, and weak acid and weak alkali. So I'll try and draw up those curves very quickly. So if I've got a data logger and I'm monitoring a solution of alkali being added to an acid, if I've got a strong acid and a strong alkali, this is the volume of alkali being added to the acid. We know that at the start the pH is very closer to 1 because we've got a strong acid. Slowly, nothing much happens at the start, and then suddenly there's a massive jump in pH. Okay, the equivalence point is about the midpoint of that graph. Then what happens as we add more and more alkali, it gets slowly more alkaline, the solution does, but not by much, okay? So what we notice is the solution is really, really strongly acidic, so loads and loads of H plus ions, and then suddenly we get to a situation where as we add more and more alkali here, not much is happening. Suddenly we get to a situation where we've got a huge number of hydroxide ions present in solution. This is when the reaction is finished. As you do a titration, what you're going to find is slowly the H plus ions are reacting with the hydroxide that's been added in, but we've got a huge excess of H plus ions, therefore the solution is always strongly acidic, and then suddenly, after we add one drop extra, suddenly we have a huge excess of hydroxide ions, and we get to this point in the reaction. Okay, so one drop of alkali is going to make a massive difference in the pH. So technically we haven't neutralized the solution, because when we add one drop of hydroxide ions, it makes a massive difference. And we've gone from a solution where we had a huge amount of H plus ions in excess. In one drop, we weren't able to completely just neutralize them. We actually had far too many hydroxide ions, and our solution became alkaline. So the pH change here that we have is going from around pH 3 up to pH 10. And if we look back at our system here, all of those indicators change color within that range, so we can use any of these three indicators for that reaction. They all change color within that range. Now if I go over to look at the strong acid and weak alkali, so I'm going to start with a strong acid in solution, slowly add my alkali via burette, and record the pH using a pH meter and a data logger. What I'm going to find is that I'm going to get up to a point where suddenly the pH jumps, because again I've added that excess of OH minus ions that I did here, so I suddenly get a pH jump to here. But because that drop contains a weak alkali, the solution itself only goes partially alkaline. So when I'm reacting a weak alkali with a strong acid, right here is where I'll find my equivalence point, but we're not going to worry too much about that. Here's where I need to worry about the range of indicators that I can use. So I can only use indicators really that are going to change color in a mildly acidic solution. If they change color in an alkaline solution, I'll have missed my point. It'll be too late. It'll be up here. So the indicators I can use for that are going to be methyl orange and litmus. So whereas I could use all three for the first one, I can only use methyl orange and litmus for this, because their range coincides with the completion of my reaction for this case when I've got a strong acid and a weak alkali. Now when we use a weak acid and a strong alkali, as we add in the strong alkali to the weak acid, we're going to get a pH change here at a certain volume, and it's going to change color at a certain range from between about 7 and 10. So we need to find an indicator that matches up with this range. So we know that the range of colors that phenolphthalein changes color at is within this range. So the only indicator I can use here is actually phenolphthalein. So if I go back up to the start, phenolphthalein is the only indicator I can use, okay, for this reaction. The reason being, methyl orange and litmus both change color before this reaction is complete. The last system we're going to look at is when we start with a weak acid and a weak alkali. It turns out when we react these two species, we don't actually get any decent range whatsoever to notice a color change. The pH rise is just so gradual that we can't, there's no indicator that will detect the endpoint of the reaction. Okay, so therefore we don't carry out titrations using weak acids and weak alkalis like ethanoic acid and ammonia, for example. Okay, so to summarize there, indicators are substances that change color in the presence of an alkali or and acid. Most indicators are weak acids, some can be weak bases, but they exist in an equilibrium between the main molecule and the dissociated form of the molecule. The dissociated form of the molecule here will have a certain color, and the molecule itself will have a certain color, or else is colorless. And if we add acid or alkali to that solution, we change the position of equilibrium, and so we change the color of the indicator. I looked at a few different indicators and their color changes and the range at which they change color, and then we looked at titration curves. Okay, these are titration curves. The 4 titration curves can be seen there. Okay, and these show us the indicators that can be used for each type of reaction. Phenolphthalein, methyl orange, and litmus, for example, can be used for a strong acid and strong alkali because their range of color change happens within the end point of our reaction. Likewise, for a weak acid and a strong alkali, we can only use phenolphthalein because its color change coincides with this range, and methyl orange and litmus we can use for a strong acid and a weak alkali, and we can't use an indicator for this color change here because the pH change is too gradual.